Hi, Assalamualaikum. So for our group, we choose Eimeria Tenella for the microbe. So actually our topic for the poster is about identification and characterization of the Eimeria Tenella microname protein. So hi, my name is Muhammad Zainuddin bin Shamsuddin. So I would like to talk more about my part, which is introduction. Microname protein, which is MIC of Eimeria Tenella, distribute over the surface of the parasite when it touch or contact with the host cells, play a key roles in the mortality, migration, attachment, invasion process. Eimeria Tenella MIC was identification and its gene structure, features, and development expression level and localization were studied. So next, these characteristics are consistent with the common features of the MIC and are named as ETMIC8. The work reported here newly identifies of microname protein of Eimeria Tenella, which is play a role in adhesive.
Based on the article, the research uses yeast called Saccharomyces cerevisiae EYB100 strain and a parasite E. Tenella strain SD01, antibodies for A7, chicken cells, and a specific type of chicken, high-line brown chicken, to investigate the inhibitory effect of traditional herbs on parasitic infection. The researchers collect and purify different types of parasite cells such as unsporulated and sporulated oocysts, sporozoites, merozoites, and gametophytes for analysis. They extract RNA from the parasite and clone ETMIC8 gene carrier region. They analyze the physical characteristic of the protein and create antibodies to it. They use different methods to analyze the protein and locate it using a fluorescent assay. The study measures the addition ability of ETMIC8 to cecum tissue and DF1 cells and tests how different dilutions of purified antibodies affect the invasion of DF1 cells. The researchers also evaluate the protective efficacy of the adhesion domains in the chickens against E. Tenella infection and measure serum antibody levels and lymphocyte subpopulation percentage in the infected chicken.
The study provides insight into the structural and functional characteristics of ETMIC8, highlighting its potential role in host-parasite interaction and its uses as a candidate for vaccine development against Eimeria infection in chicken. There are various results obtained from the observation, and I would like to highlight some of the results. The protein level of ETMIC8 in different developmental stages of E. Tenella were examined by Western blotting. Results shows that ETMIC8 expressed in unsporulated oocysts, sporulated oocysts, sporozoid, merozoid, and gametophyte of E. Tenella can be recognized significantly by anti-ETMIC8 MAB shown in Figure 2I. The ETMIC8 protein expression levels were 2.97-fold higher in sporozoid, 2.4-fold higher in merozoid, and 3.88-fold higher in gametophyte than in unsporulated oocysts shown in Figure 2G.
Okay, hello, my name is Muhammad Daniel and I will be presenting on the first part of the discussion. Now, I will begin. ETMIC8, which is identified as a critical protein within the Eimeria Tenella parasite, assumes a pivotal role in the intricate dance between the host and parasite during the infection. Its significance stems from possession of the EGF domains, which is the epidermal growth factor, which is acting as molecular keys that facilitate the parasite's attachment and subsequent penetration to the host cell. Basically, it is an important part of how the parasite managed to get into our system. So this is an indispensable step for the parasite survival and proliferation within the host organism. This ability to mediate invasion underscores its fundamental role in the parasite's life cycle. But then, at the same time, ETMIC8's interaction with the host elicits a substantial immune response. This dual functionality is like a double-edged sword, because on one side it helps the parasites persistence, but on the other side, it also triggers the host immune system response. And that's all for me. Thank you very much.
So hi, my name is Abdul Aziz bin Ismail, and I'll be continuing on the discussion part. The delicate balance between helping invasion and sparking the immune system illustrates the intricate relationship between a parasite's survival tactics and a host defense mechanisms. Exploring these interactions not only unveils how parasites invade and stay in hosts, but also identifies weaknesses that could be targeted for interventions against Eimeria Tenella infections. This understanding opens the door for developing focus on treatments or vaccines to disrupt invasion or use the immune system to fight the parasite survival tactics.
So last but not least, I would like to talk about conclusion. So basically, this study discovered ETMIC8, a novel microneme protein in Eimeria tenella. So ETMIC8 with a typical microneme features show developmental stage, specific expression, and play a key role in adhesive and invasion. So in this study also discovered anti-ETMIC8 antibodies that can reduce invasion rate, and the ETMIC8 EGF peptide displayed a moderate protective efficacy, suggesting its potential as an immunogenic target for the subunit vaccine. So basically, these findings advance our understanding of Eimeria tenella invasion and offer insight for vaccine development.